So, hello everyone, good evening. Welcome to today's live session of week 8 Immunology. I am Surasree, PMRF scholar of IIT Kharagpur. And today in this session we will be discussing the several steps of maturation, activation and differentiation of B cells or B lymphocytes. Okay, so in the previous session we were discussing about how T cells are matured, T cells are activated, right? So now in this session we will be discussing about B lymphocytes and how the B lymphocytes which originate in the bone marrow, how do they become matured, and then how do they become activated when they come in contact with antigens, right? So in case of T cells also we had observed this, that in case of T cells also, the naive T cells, they become activated or they become effector T cells when they come in contact or when they encounter antigens, right? So similarly in case of B cells also we will see the similar phenomena. But before going into that, let's first see how the B cells, the B lymphocytes, the progenitors of B lymphocytes, how they are activated, how they are matured, how they first mature to form the B cells which can produce antibodies, right? Okay, so welcome to today's session. Let's start. Okay, so firstly we are going to discuss about B cell maturation, and we know that B cells, the population of B lymphocytes, they mature from the hematopoietic stem cells in the bone marrow, right? So they originate in the bone marrow. Right? They originate in the bone marrow and they first form these multipotent progenitor cells. The multipotent progenitor cells will finally give rise to the lymphoid progenitor cells. So from the name lymphoid progenitor you understand that these cells are destined to become lymphocytes, either B lymphocytes or T lymphocytes or maybe NK cells also, right? So these from the lymphoid progenitor, further the pro B cell, and from the pro B cell the pre B cell, and finally the B cell, the mature B cell will be this maturation process will take place, right? So first let us look into what is the molecular mechanism by which these multipotent progenitor cells finally become immature B cells, right? What is this process of maturation? So you can see here, this particular cell is a MPP. What is an MPP? MPP is multipotent progenitor cell, right? So an MPP, when it comes in contact with the stromal cell inside the bone marrow, there is a ligand which is expressed on these stromal cells. The ligand is FLT3L. L stands for ligand. What is FLT3? FLT3 is a tyrosine kinase receptor. Okay, so this tyrosine kinase receptor is expressed on the multipotent progenitor cells, and this FLT3 receptor can interact with this FLT3 ligand on the expressed on the surface of the stromal cells. So once this interaction takes place, the multipotent progenitor cells comes in contact with the stromal cell, right, through this through this interaction of the cell adhesion molecules. Next, what happens? This multipotent progenitor cell will become the lymphoid progenitor cells, right? And in this process, there is one interleukin, right? Interleukin is a cytokine. You know that interleukin 7 is involved in this process. So these molecules here are interleukin 7, which are expressed by the stromal cells. Okay, Whereas the interleukin 7 receptor, this particular receptor, the interleukin 7 receptor, is present on the multiprotein progenitor cell. So once this kind of interaction, the interaction between the binding or the binding of IL-7 to IL-7 receptor takes place, the multiprotein progenitor cells become destined to lymphoid origin, right? So they become the lymphoid progenitor cells. Now, from these lymphoid progenitor cells, the cells which will be destined to enter into the B cell lineage, they will express several cell adhesion molecules and several receptors on their surface, right? So let us look into what are these receptors. The first one is VCAM1 VLA4. Okay, so I'm writing it here. VCAM1 VLA4 binding. VCAM1 VLA4, I hope you remember from the previous session that these are the integrins, right, or the cell adhesion molecules which help in the interaction. So when we were discussing the interaction of T cells with antigen presenting cells, we were also discussing this type of cell adhesion molecules and integrins, the role of these integrins. So these integrins are also important while the B cell is developing, right? So the first one is the interaction between the binding of VLA4. VLA4 is present on these pro B cells, or you can say the lymphoid progenitor cells, right, which is now going to develop into pro B cells, and VCAM1 is present on the stromal cell inside the bone marrow, right? Once this binding of VLA4 to VCAM1 takes place, there is another receptor which is expressed on these pro B types of cells, or progenitor B cells, right? Pro stands for progenitor. Okay, that is c-KIT and SCF. c-KIT is the tyrosine kinase receptor. c-KIT is again another tyrosine kinase receptor. Okay, and what is SCF? SCF is stem cell factor. Okay, so this SCF is expressed on the stromal cell surface, and the c-KIT molecule, which is a tyrosine kinase receptor, the binding of this stromal stem cell factor to this c-KIT receptor again induces several changes, or it actually starts this signaling cascade by which this progenitor B cell will now be able to mature into pre-B cells or premature B cells. Okay, and there are several other processes which are also taking place while the cell is maturing from lymphoid progenitor to progenitor B cells. So what is this? What are these processes? So the arrangement, the rearrangement and the recombination of the V chains, VDJ recombination, right? VDJ chains. So this is taking place inside these progenitor B cells. So there are separate phases in which these recombinations are taking place. So in the next slide we are going to discuss in detail about that. But just before going into that detail, we are now discussing the factor which is actually helping these B cells to develop, right, from progenitor B cells to premature B cells to immature B cells and finally to mature B cells. Now, so you can see on the pre-B cells, so once these interactions, the VCAM1 VLA4 and the c-KIT SCF interaction, the binding of them has taken place, these pro-B cells have developed into or matured into pre-B cells, pre or premature B cells, right? Now these premature B cells, why are they called premature? Because they express a surrogate light chain, and so in the immunoglobulin molecule, you know, in the antibodies, right, there are two chains, the heavy chain and the light chain. But when the B cell is still in the premature B cell stage, then the light chain rearrangement has not been completed. So they express a surrogate light chain on their surface, and they also express the rearranged heavy chain. Now in this particular phase, one important event takes place, that is the binding of IL-7 to IL-7 receptor on the pre-B cells. Right. So you can see here, first of all, these pro-B cells, the lymphoid progenitor cells, they were bound to the stromal cell surface by the interaction of several cell adhesion molecules and several receptor molecules, right? So now the pre-B cell which are developing, it will move away from the stromal cell, or their interaction or the binding between the stromal cell and the B cell will dissociate. For that, this binding of IL-7 to the IL-7 receptor has a very important role. How? So this is the IL-7 molecule and this is the IL-7 receptor, right? Once this binding of IL-7 to IL-7 receptor takes place, the cell surface molecules which were helping the pro-B cell to adhere to the stromal cell surface, those expression of those cell surface molecules is going to be down regulated. Okay, so the expression of VLA4, for example, is going to be down regulated, and because of this down regulation, what actually happens is that this adhesion, the cell adhesion, is destroyed, or it is affected, right? Because of which these pre-B cells will now be able to move away from the stromal cells, and it will finally mature into the immature B cells, right? So these immature B cells now express IgM on their surface, immunoglobulin M. And remember that in the immature B cells, the rearrangement of the light chain and heavy chain has both been completed, because of which they have become now immature B cells. Now these immature B cells will again undergo several changes by which they will become mature B cells. Okay, by which they will become mature B cells, and then finally it will enter the bloodstream. Right? So this is the overall process how the maturation of B cells take place. Now we will look at what are the events, specific events which are taking place inside the B cell, because you know that the function of B cell is to produce these immunoglobulins, right? So when the B cell is maturing, there will be several changes in the immunoglobulin molecule, which is expressed on the cell surface, right? So or which is called the membrane immunoglobulin molecule, right? So which is why there are several events which will go inside the B cell, and now we will have a look at that. So this slide talks about what are these specific events which are occurring inside the B cell while the B cell is maturing. So here you can see this is the stem cell with which we have started, the multipotent progenitor cells, right? Now when they become lymphoid progenitor, and after which they become the pro-B cells or the progenitor B cells, in the progenitor B cells there are two separate stages. One is early progenitor B cells and the other is late progenitor B cells, right? What is happening here? So inside the progenitor B cells, the rearrangement of the heavy chain is taking place. So this is heavy chain rearrangement, right? I'm writing it here, pro-B cell, heavy chain rearrangement is taking place. And most importantly, in the early progenitor B cell, in the early progenitor B cells, the D to J, DJ rearrangement is taking place, while the progenitor B cells are in the late phase of their development, the V to DJ recombination is taking place, right? So you know that in the immunoglobulin molecule, these two types of recombination events will take place while the heavy chain is being rearranged, right? So DJ recombination and then VDJ recombination. And so once this has been finished, once the VDJ recombination is completed, so that means the heavy chain rearrangement is done, right? No more changes will be occurring in the heavy chain. Now what will happen is that these late pro B cells, these late progenitor B cells now will express this heavy chain on their surface, okay, along with Ig alpha, and there will also be an expression of Ig alpha and Ig beta. What are these Ig alpha and Ig beta? Can anyone tell me? These
The co-receptors.
Actually they are co-receptors, but they are actually the signaling component of the immunoglobulin molecules, okay? So they are responsible for signaling, because when the immunoglobulin is membrane bound, the binding of any antigen to this membrane bound immunoglobulin must initiate some signal or must provide some signal to the B cell so that the B cell can take further actions, right? So this signaling component is comes from the Ig alpha and Ig beta. So it actually looks like this. So this one is the Ig, right? And these two are the Ig alpha and Ig beta. All right. So they provide the signal once there is some change, once there is some binding in the immunoglobulin molecule. All right. So the late progenitor B cells have now completed their VDJ recombination of the heavy chain, and they are also expressing Ig alpha and Ig beta on their cell surface. Now once these progenitor B cells will further mature to become premature B cells, there this expression of surrogate light chains are you can see the surrogate light chains on these pre-B cells or the premature B cells. Why? These surrogate light chains are made up of the two chains which are V pre-B and lambda 5. So these are the two chains which are also polypeptide chains, which are the surrogate light chains, which act as the surrogate light chain. Why is this surrogate light chain being expressed? Because there has not been the light chain rearrangement has not been completed yet, right? So this light chain rearrangement is still taking place, right? Is still taking place in the premature B cells, which is why the surrogate light chain is being expressed with the heavy chains and along with the Ig alpha and Ig beta. All right, this is the immunoglobulin with heavy chain rearranged, but surrogate light chains are expressed, and also the Ig alpha and Ig beta are expressed, right? So this is the premature B cell. Now once we come to the immature, so these premature B cells will now further develop to form the immature B cells. Now I told you that in the immature B cells the light chain rearrangement will be completed. All right, so here you can see that the immunoglobulin molecule is expressing both heavy chains and light chains along with these signaling components Ig alpha and Ig beta. So this population is the immature B cells, and it expresses only IgM on their surface, right? So these are all membrane bound immunoglobulins. Now further these immature B cells will further mature to become mature B cells, which will be expressing two kinds of immunoglobulin molecules, IgM and IgD, right? So this is how the B cell maturation, and what these are the events which take place inside the B cell while it is maturing. And one important point here is that while these B cells will mature, right, from the immature to the mature B cells, what happens, what are the changes which occur inside the B cells while it changes from immature to mature B cells. Now let's have a look at that. So there are 4 different fates which can occur to the immature B cells. So the immature B cell is expressing heavy chain and the light chain, right? The rearranged heavy chain and the rearranged light chain of the immunoglobulin. Okay, so one possibility may arise that these B cell receptors are being able to recognize and react to the self antigens, right? So this kind of selection was also taking place while we studied the T cell maturation process, right? So the T cells which are reactive to the self antigen, they are usually deleted, and they are so they die by apoptosis. We saw that, right? So therefore the similar phenomena has to occur to the B cells also, which react to self antigens, because the immune system will always try that the cells are not responsive to our self antigens. Alright, so depending on whether they recognize and react to the self antigens or not, there are several fates which can occur to the immature B cells. Now let's see what are these fates. One is that this, for example, number 1, what is happening here is that the immature B cell is not being able to recognize any self antigen. So I'm writing it here. Immature B cell does not react to self antigen. So what do you think will be the fate?
It will mature.
Yes, this population will be selected, right? So they will develop into mature B cells. All right. Next is the opposite of this, that is the B cell, immature B cell, is reacting to the self antigen, reacts to the self antigen. So if that happens, there will be 2 events which can occur here. One is that the immature B cell might die by apoptosis, okay, or there is another event which can take place here, which is receptor editing, which means that this particular receptor is being able to recognize self antigen and react to it, right? So if I rearrange this particular receptor once again, the receptor might be able to might be able to not react to the self antigen, right? So there will be several rounds of receptor editing which can take place in this particular stage, right? If the B cell receptor which is expressed on the immature B cell is being able to react to the self antigen, right? So either the cell will die by apoptosis, or it may also undergo receptor editing, in which further rearrangement of the heavy chain and the light chain will take place. Alright, and after this rearrangement, this population, so suppose this is the cell which has undergone this receptor editing, and so it will have, this particular receptor will have new receptor specificity, right? And again there will be this selection process, that whether this particular receptor is now being also being again able to react to self antigen or not, and if it does not react to self antigen, then it will again mature, right? So if it does not react to self antigen, it will mature. Otherwise, if it again reacts to self antigen, another round of receptor editing might take place, or if the cell thinks that this particular receptor will not be able to will always react to self antigen, then the cell will die by apoptosis. Okay, so this kind of receptor editing also takes place. So if it reacts to self antigen, there are two fates which may occur. One is apoptosis and another is receptor editing. Okay, coming to the third point, which is, suppose these immature B cells, they interact with a soluble antigen. Okay, so they can bind to a soluble antigen, and once they bind to soluble antigen, what happens is that there is a cross linking of the receptors which takes place. Cross linking of receptors takes place, because of which what happens that these immature B cells become unresponsive to any antigen, and this stage is called anergy. This phenomenon is called anergy. Okay, anergy is something which happens when the immature B cell is being able to react to any soluble self antigen, right? Which is why these cells are actually, they cannot interact with antigens anymore. So they enter into this state of anergy, right? And the last fate, the last thing is that if the immature B cells, right, the immature B cells are being able to react to foreign antigens, but they are interacting with foreign antigen with a very less affinity. So here, in case of, in this point, fate 4, there is something which is important, which is less affinity. I'm just writing the point 3 once, that is reacts to self antigen soluble, enter into the state of anergy. And number 4 is reacts to foreign antigen with less affinity. And if that happens, this population will be clonally ignorant. So that means they will still mature, but they will not be able to interact, or they will not be allowed to interact with any foreign antigen. Because why? Because they are interacting with foreign antigen with a very less affinity, which is not very good situation. Because if there is a foreign antigen, if there is some pathogen and the B cell is interacting with a very less affinity, then it might not induce the signal which is required to eliminate that foreign antigen, right? Which is why this population becomes clonally ignorant. Okay, so they still mature, but they are not allowed to interact with the antigens anymore. So these four are the fates which can happen to an immature B cell while it becomes from immature to mature B cells, right? So does anyone have any question till this?
These mature B cells will be called as naive B cells, is it?
Yes, but actually these naive cells, this term is actually mostly used for T cells. So these B cells are called mature B cells.
Okay, ma'am. Thank you.
What is the function of immunoglobulin when it is present on the surface of B cells? It has the same function as that of the antibodies. When it is present on the surface of the mature B cells, so they also interact with antigens, right? And so actually what happens is that whenever there is any infection or anything, like these cells which are expressing the immunoglobulin molecules on their surface, they come in contact with the antigen, or they go on to find if there is any foreign antigen or not. And if there is something like if they interact or if they bind to any foreign antigen, then what happens? Then the B cell, we will see also later on, the B cells become activated. Okay, and once they are activated, they can either become a memory B cell or a plasma B cell. Okay, the plasma B cell will now be producing soluble antibodies. So these cell surface molecules which are being able to recognize this foreign antigen, they will now be secreted in soluble form. Okay, but the point is that all the surface immunoglobulins, they have the same function as that of the antibodies. So they bind to the antigen, right? Okay, cross-linking. Cross-linking means these receptors. So whenever there is a multivalent antigen, so suppose the multivalent antigens means what? Suppose they have several epitopes on the surface. So once these several epitopes are there, so it can be recognized by several receptors on the B cell surface, right? So like this one. So this particular antigen is being recognized by this receptor as well as this one, right? So which means that the receptors on the surface are cross-linked together by binding to the same antigen molecule. Okay, there was another question. Is that okay? Nishtha is asking, can you please tell the difference between the second and third fate? Yes. So the second fate is concerned about the cell surface antigens, right? So these are the cell surface antigens. Present on the surface of the self cells, right? And if the cells are if the B cells are reacting to the cell surface self antigens, then either they will die by apoptosis or they will undergo receptor editing. And the number 3 is concerned about the soluble antigen. So if there is a soluble antigen which is this B cell is being able to recognize self antigen, then it will undergo this energy set. That means there will not be any further maturation of this immature B cell. Okay, the maturation process will stop there, and they will later on die. So the fate finally is death only. But they will, for the time being, they will enter into this state of energy. Soluble antigen means which is soluble, which is not bound to the cell surface, which is soluble in the bloodstream, or which enter into the lymph nodes, right? When B cell recognize antigen, does it send signal to cell mediated immunity, or it will involve in immune response by itself? So once the B cell recognize any antigen, there can be many things that can occur. One is opsonization, right? We were discussing neutralization, opsonization, and also there can be other events where the B cell will deliver its antigen, where it can work as an antigen presenting cell also, right? And it can deliver its antigen, the antigenic peptide, to the T cells, to the T helper cells, where the T helper cells will again function in stimulating the immune response. So there can be multiple pathways by which the B cell will try to eliminate that pathogen. If you remember, initially in the previous sessions we discussed about these opsonization, neutralization, and these things. Is that okay? Okay. So if you do not have any other questions, let's move on to the next slide, where we will be discussing how these mature B cells will now become activated. And these activation process, actually there are two types of there are two separate ways by which these B cells can become activated. So one is a thymus dependent process and another is a thymus independent process. So let's have a look at that. So these here in this section you can see that this maturation event has taken place inside the bone marrow, right? Now after these cells are mature, that means after the B cells which does not react with self antigen, after they are selected, now they come out of the bone marrow and now they migrate to the peripheral lymphoid organs or the lymph nodes, right? Now once they come to the lymph node, the lymph node has several sections: the paracortex region, the cortex region, the medulla region, right? And inside the cortex there are centers which are called germinal centers. So in these germinal centers the B cell activation process takes place. So this process is just an overview. So the T cells will now become the B cells will now become activated, right, once they come in contact with an antigen, and then finally these B cells will be differentiated. So the two processes will take place one after another. First is maturation from the progenitor B cell to the mature B cells, then the activation of the B cells where they will encounter antigen, and finally after an encounter of antigen they will become differentiated so that they can perform its designated functions, right? They can become plasma cells or they can become memory B cells. So this B cell activation process takes place, I told you, that in two different pathways, a thymus dependent and thymus independent process. So in thymus independent process, which means thymus independence means you may understand that it is independent of the T cell interaction, right? Thymus means that the T cells are matured in thymus, so of course the the help of T cell is not required. So this is called TI, thymus independent process. Here what happens, the pathogens or the suppose bacteria or any other microorganism which have on the cell surface several antigens are present on the bacteria, right, which can be recognized by the B cell receptors. So if the B cell receptor, this is the B cell receptor, right, immunoglobulin molecule, this directly interact or directly bind to the antigens which are present on the surface of the pathogens. So then what happens, there is a signaling that takes place, the signaling that I was telling you about Ig alpha and Ig beta, right? So once this BCR binds to this antigen on the cell surface of the pathogen, right, so these antigens can be LPS, right, lipopolysaccharides which are present on the cell surface of bacteria, or it can be the bacterial DNA fragments, right? There can be several other antigens also. So once the BCR binds to this particular antigen, what happens, this Ig alpha, Ig beta starts a signaling through its transmembrane receptor, right? So it has a so this Ig alpha, Ig beta molecules, they have cytosolic part and they also have the extracellular part of this chains, right? And in the cytosolic part there is this motif ITAM, immunoreceptor tyrosine associated motif. Item. So these motifs are present on the cytosolic domain. Sorry, do you have any query? Okay, once there is this signaling, the signaling is actually induced by this item motif, right? So this induces this signaling, and this signaling will help to activate the B cell. Okay, and this thymus independent process actually can have two subtypes, this type 1 and type 2. In the type 1 there is no involvement of any T cell, right? So the bacterial the antigens which can be recognized by these by the B cell receptor are the LPS, the bacterial DNA, like these, okay, which are present on the pathogen surface, pathogen cell surface, and which are directly recognized by the B cell receptor. Whereas in case of this type 2, there is involvement of T cells, not by direct interaction with the B cell receptor, but by the help of the cytokines which are produced by the B cell receptors. Cytokines which are produced by the T cells. Okay, so there is no direct involvement of the T cells, but the help of the cytokines are taken. And here there are some repetitive structures, repetitive structures on the pathogen cell surface which are recognized by the B cell receptor, directly by the B cell receptor, but in this case of activation there will be involvement of some cytokines which are secreted by the T cells, right? So this is the thymus independent process, and in the thymus dependent process, you know that this is called TT. So in the thymus dependent means it is dependent on the T cells, right? So here you can see that this is an antigen molecule, right? This antigen molecule, the B cell receptor on the surface of the B cell can recognize this particular antigen molecule, right? And once this recognition takes place, there is again this cross-linking of the receptors that take place, right? And due to this, once this takes place, you know that the B cells are the antigen presenting cells, so they can phagocytose foreign pathogens, right? So this cross-linking of the receptors with the antigen actually helps the B cell to phagocytose this entire complex, right? And after this phagocytosis, the foreign pathogen is degraded. So remember this phagolysosome that forms where the foreign pathogen is degraded, right? And then what happens, this foreign peptide comes in contact with the MHC class 2 molecule. So this is a MHC class 2 molecule which will now form a complex with this foreign peptides, right? And now this MHC class 2 molecule will express this foreign peptide on its surface like this, right? So the MHC class 2 molecule will now express this foreign peptide on its surface, which can now be recognized by a T cell receptor. Okay, and what is this particular T cell? This is the T follicular helper cell. So this T follicular helper cells, in the last session we were discussing about the several effector T cells, and T follicular helper cell was one, and I told you that the T follicular helper cell is responsible for interaction with B cells and for activation of antibody productions and all, right? Class switching, these events are brought about by the interaction of the B cell with the T follicular helper cell. So here is this interaction taking place. The B cell is expressing the foreign peptide on its surface with the help of this MHC class 2 molecule, and it is being recognized by the TCR. And of course in this interaction there are several other co-stimulatory molecules, right? So which is B7, CD28, these things we discussed in the previous session also. if you try to remember. Also the interaction of CD40 ligand and CD40 on the T follicular helper cell and the B cell. By this kind of co-stimulatory interactions, and also with the help of the co-receptor, which is the CD4, right? So the MHC TCR interaction is taking place, because of which now this T follicular helper cell will produce an interleukin, which is interleukin 21 or IL-21, right? Because of this interaction with the B cell with the T follicular helper cell, the T follicular helper cell will now secrete this important cytokine IL-21, which will further initiate the JAK-STAT signaling. Okay, what is JAK-STAT? STAT is a transcription factor. And JAK is a kinase, right? So because of this expression of IL-21, it induces this JAK-STAT signaling, and it actually induces this expression of STAT3, by which it induces the signaling of JAK-STAT, and this signaling helps it will now help the B cell to proliferate, right? So what is the event taking place? First is BCR recognizes foreign antigen. Next is phagocytosis of the foreign antigen. I'm just summarizing the whole event. Next is expression of this foreign antigen by MHC class 2. On B cell, on the B cell surface. Next is MHC class 2 TCR interaction. Which helps in increased production of IL-21, and the IL-21 helps to induce the JAK-STAT signaling, which will further help the B cell to divide and proliferate. So this is the total process that takes place, and after these events are successfully over, the B cell population will proliferate in the germinal center, right? And it will express some receptors on its surface, some chemokine receptors. What is the role of these chemokine receptors? We will see in the next slide. But once these B cells are activated in the germinal center, they will now undergo differentiation to become either the plasma cells. So the plasma cells, what do? They will now secrete these immunoglobulin molecules. They will produce these immunoglobulin molecules in much higher quantity, and now they will secrete them so that these immunoglobulins can reach in the bloodstream and then can neutralize the toxin, or then they can also do the opsonization, right? Or they can become they will also become memory B cells, which will help the body to fight any further infection by the same pathogen, right? And these memory B cells will go to the bone marrow for any future use. All right, so this is how the B cell activation is taking place. Next, if we come to the differentiation process, so these activated B cells, I told you that these activated B cells, they express this receptor, chemokine receptor CXCR4 on their surface. What is the role of this receptor? The CXCR4 interacts with this ligand CXCL12. CXCL12 is nothing but a chemokine, okay? And this interaction of this chemokine with this chemokine receptor retains these centroblasts. So these are called the centroblasts, or the activated B cells. retains them in the germinal center. So the germinal center has two zones. One is the dark zone and the light zone. So the activated B cells are still in the germinal center dark zone, right? And here further events will take place which will now help the B cells to differentiate and become a better, more efficient B cell. Okay, so how is this efficiency induction, how is this going to happen, like how are they going to become more efficient? So there are two events which will take place in the germinal center. One is somatic hypermutation and the other one is class switching, right? What is somatic hypermutation? This is taking place in the dark zone of the germinal center, where the centroblasts or the activated B cells, they will undergo mutation in the variable region of their B cell receptor. Okay, so somatic hypermutation is random mutations in the variable region of their receptor of BCR. Okay, so this somatic hypermutation is induced by this enzyme. If you remember the name of this enzyme, AID, activation induced
deaminase.
Activation induced cytidine deaminase. So what does it do? It first actually cleaves off the cytidine, and which induces several DNA repair processes. And these DNA repair processes can be like base excision repair, DNA mismatch, like mismatch repair. Because of these repair processes, what will happen? There will be some mutations which will be introduced in this particular DNA fragment, right? And because of these mutations, there can be several things which can happen. Either the affinity of the B cell receptor will increase, or the affinity of the B cell receptor might decrease also, because these are random mutations and these are taking place. So obviously there is a chance that it will increase the affinity of the B cell receptor, or there is a chance that it can decrease the affinity of the B cell receptors, right? So now the T cells which will have only increased affinity towards the B cell receptor will be selected after this somatic hypermutation event. Okay, and how is that taking place? First of all, this somatic hypermutation has taken place in the dark zone, right? Because of which there are several centroblasts or several activated B cells are produced, which are having now different B cell receptors, right? Because of the mutations that has taken place. And now these centroblasts or the activated B cells will now migrate to the light zone of the B cell of the germinal center. Okay, how is this migration taking place? It is mediated by this by these two chemokines and chemokine receptor. CXCL13 is the chemokine and the CXCR5 is the receptor for the CXCL13. Okay, so since the activated B cells now they express CXCR5, so they will be attracted towards the light zone, where this CXCL13 is expressed by these follicular dendritic cells. So these follicular dendritic cells, they reside in the light zone, right? And they express this CXCL13, which attracts the centroblasts to the light zone. Now once they reach the once they reach the light zone, they are called now centrocytes. So now these B cells can have either high affinity or low affinity for the foreign antigen. So now therefore there has to be another selection round that has to take place here, which will select the B cells with respect to its affinity, right? And this process is nothing but affinity maturation, so which is taking place in the light zone of the germinal center. Okay, and this affinity maturation is nothing but selecting the B cells which have higher affinity. Okay, and the low affinity B cells will finally die by apoptosis. Okay, and the last event, which is now going to take place after this affinity maturation is the event of class switching. So what happens in this particular stage, that is the B cells which have this high affinity, so they will express the foreign antigen, so they can interact with the follicular dendritic cell, and if they have this high affinity, they are going to be selected. Fine. Once they are selected, now they will internalize the antigen, and they will again express it with the help of this MHC class 2 molecule, right? Now this foreign peptide is being expressed by the MHC class 2 molecule, and the T follicular helper cells with the help of its TCR can recognize this MHC class 2 molecule associated foreign peptide, right? And once that happens, once this T follicular helper cell recognizes this antigen on the surface of the B cell, there are several cytokines which come into action now. So these cytokines are expressed by the T follicular helper cells only, and depending on the type of cytokine, the B cell will now differentiate into several classes of B cells. So that means they will now express several classes of antibodies like IgE, IgG, right? So for example, so this is just a brief overview of what kind of cytokines can be there. So IL-4 will induce the expression of IgE, right? And IFN gamma, INF gamma, and your IL-21, they will also induce the expression of IgG molecules, right? So depending on which type of cytokine is being produced, the B cell will be able to switch its class, right? Or it can or it is known also as isotype switching. So there will be rearrangement in the heavy chain, in the constant region of the heavy chain, right? Because of which it will express several classes of immunoglobulin molecules, and these B cells will now be differentiated into several classes, right? So this is how the activation process and the differentiation process takes place in the germinal center of the lymph nodes. Now once these T cells are totally differentiated, they will now become either plasma cells or memory B cells. Now the plasma cells will enter the circulation, or the memory B cell will enter the bone marrow, right? So these are the few events that you need to remember. One is somatic hypermutation, which is taking place in the dark zone, right? So the mutations are occurring on the variable region, of the B cell receptor. Next, these B cell receptors which will have high affinity for the foreign antigen will be selected by affinity maturation, and finally the B cell will undergo class switching, where it will be expressing several classes of antibody molecules depending on which type of cytokine signal is present, right? So please tell me if there is any doubt.
I was going through the Cube in that book. B7 is not given as the one interacting with CD28. They've given something else. I think they have given CD80 oblique CD86.
Yes, that is actually renamed as B7 only.
Okay, thank you, ma'am.
CD86 is same as B7, and you will see that I think CD80 is written as this B7.1 and 86 is written as 7.2, something like this.
Okay, ma'am.
Yes. So please let me know if you have any other doubts, or we will now straight on go to the question answer session.
Ma'am, one question.
Yes.
So these cytokines, they are responsible for the IgE, G or whatever, right?
Yes.
So they are present on the plasma cells, okay, which are released. So what about the memory cell? I didn't get this point. How they will get converted into memory cells? So they won't have these IgE?
No. Of course they will have. So either the particular B cell, so if you take in consideration one particular B cell which is interacting with the TCR, right, with the help with the T follicular helper cell. So this particular B cell will either produce IgE or IgG or other types of immunoglobulins, right? So suppose it is expressing the T follicular helper cell is giving IL-4 signal and it is being able to express IgE. Okay, so the memory B cell will express IgE on its surface, but the plasma B cells will express the soluble form of this IgE. Okay, so which will be coming in the circulation, and it can directly interact with the antigen.
Okay. Means both memory cells and plasma cells have IgE, but in case of plasma cell, it will be released.
Yes, it will be in the soluble form.
Okay, but on memory cell, it is still be there.
Yes, IgE will be expressed because you need to retain that memory that this pathogen had come into my body, and this is the way I have to fight this pathogen, by expressing IgE. So that memory has to be retained in the bone marrow, and that will be retained by the memory B cell.
Okay, ma'am. Thank you so much.
Okay.
So, ma'am, can we call these as the free antibody and the membrane-bound antibody?
Yes. So the surface immunoglobulins are often written as this MIG, or the membrane-bound immunoglobulins, right? And the soluble immunoglobulins are written as antibodies, or they are termed as antibodies, right? Okay. Any other doubt? If not, then let's go to the questions. Okay, tell me your answers in the chat box. Yes, your answers are correct. It is the activation induced cytidine deaminase, right, the enzyme which is responsible for somatic hypermutation. Now, which cell is the connecting link between innate and adaptive immune system? No, most of you are actually wrong. Yes, the correct answer is D, dendritic cells, and not the B cell. Don't be confused, because B cell have these antigen presenting properties. It does not come under innate immune system, right? It is totally inside the adaptive immune arm of our body. So which is why the connecting link between the innate and the adaptive system is dendritic cells. Okay, the dendritic cell is the correct answer. Okay, question number 3. What is affinity maturation? So all of you who are saying D, all of the above, scan all the options carefully. So here the correct answer is C, because the B cells which have the higher affinity for foreign antigen, they are selected, whereas the B cells which have low affinity for the foreign antigen are deleted. This is not actually affinity maturation. they die by apoptosis. So the affinity maturation is correct for this particular incident, that is the B cells which have the high affinity for foreign antigen are selected, and this is completely wrong. The B cells which have the high affinity for foreign antigen are deleted. This is wrong. So all of the above is also wrong. So the correct answer is C. B cells having high affinity for antigen are selected. This particular question is a little bit complex. Please go through it carefully. The steps for maturation of progenitor B cells to precursor B cells. What is the correct sequence of events? Okay, so the correct answer is B. Okay, the correct sequence, let's just figure it out once. So 3 comes in the first place. So binding of VCAM1 on the stromal cell and VLA4 on the pro B cell. That is the first event that takes place. Next, it is followed by the binding of the c-kit to the stem cell factor. Right? So these are the two first events, 3, 1. Then next comes the expression of the IL-7 receptors on the pro B cell, which is followed by binding of the IL-7 released from the stromal cells to the IL-7 receptors on the pro B cells, right? Which will actually help the pro B cell to come away from or dissociate from the stromal cells, right? The cell adhesion molecules will now be released, and then it will finally mature from pro B cell to pre B cell. So this is the correct sequence: 3, 1, 4, 2, 5. Right? That is the solution. Does anyone have any doubt in this question?
No, ma'am. Thank you so much.
Okay. Okay, if not, let's move on to ask this question. Somatic hypermutation, where does it occur? Yes. Yes. Your answers are correct. Somatic hypermutation occurs in the dark zone. Yes. Okay. So last question for today. This question was there in assignment 7, question number 3. Someone asked me in the asked me the discussion forum. So the question was, this is from assignment 7. Question number 3. The question was, the thymus from a skid mouse was planted in a nude mouse. What will be the difference in the spleen cell population? Can anyone tell me the answer, or yes.
Number of mature T cells will increase, ma'am.
Yes. So can you explain, Vandana?
Ma'am, in the nude mouse, there is the thymus have been removed.
Yes.
But the bone marrow is functional. So both the B and T cells will originate and the B cells will mature normally. The only defect lies in the maturation of the T cells.
Yes. So
And
no
if such a
mature T
nude
cells.
mouse, there is planted, their thymus is planted in such a nude mouse, then even the T cells will mature normally.
Okay. And can you elaborate like what is a skid mouse?
Severe combined immunodeficiency mouse. In this mouse, both the B and T cells are deficit.
And what is the reason behind that?
And
So what happens in this skid mouse actually? It's a severe combined immunodeficiency disorder it's suffering from, and the deficiency is actually lies on a protein kinase. So this is very interesting. It has all the thymus and bone marrow, everything is normal, right? But the deficiency lies in this particular enzyme, protein kinase, which is involved in the VDJ rearrangement. Okay. So rearrangement of T cell receptor and B cell receptor is, so this enzyme is deficient, so of course no rearrangement of TCR and BCR takes place, right? Which is why the B cells and the T cells are deficit, or the B cells and the T cells are not active in this kind of a mouse. Okay, so SCID comes from this deficiency or the inactivation of the protein kinase enzyme. So of course in a SCID mouse, so the question was actually that in the SCID mouse, both the B cells and the T cells, they are not active, or they are not activated, they cannot mature. So how will the thymus of that SCID mouse, if it is transplanted to the nude mouse, how will it help? But the problem actually lies in this part, that the SCID mouse, it has everything normal from the thymus to bone marrow, but the deficiency lies in the protein kinase enzyme. So if the thymus is taken out from the SCID mouse and planted in the nude mouse, which does not have any thymus, then of course this maturation of T cells will take place in the thymus, and therefore the number of mature T cells will increase. So that is the correct answer here. Is that okay?
Yes, ma'am. Very well explained. Thank you so much, ma'am. Ma'am, I have one more query. Can I put up now, or you have more questions to give?
Yeah, you can ask me now also. No problem.
Ma'am, the JAK-STAT, is this the mechanism of action of insulin hormone as well?
Yes, absolutely.
Thank you so much, ma'am.
Yeah. So these signaling components, if you are interested to learn, you can go through the Albert's cell biology book.
Okay, ma'am. Thank you so much, ma'am.
Okay. So this is the solution, and please let me know if you have any other queries, or we will end the session here. Okay. Okay, thank you for joining today. So next session we are again meeting next Tuesday 7 p.m. If you have any doubts, please let me know your queries in the forum, and also you can ask me personally. And so let's end this session today here. Okay, bye.